Uh, got it. One of my oldest friends as a grown up and as a researcher. So, and in fact, one of my primary reasons for being here is the influence of Steve Cole, and he knows this, but he probably forgets this. So Steve Cole is a professor at UCLA in neuroscience and psychology, and he has had the most interesting and amazing career. We're so obsessed with his research. We basically call him and collaborate with him and get his advice on almost all our studies. Eric and I are, let's just say, eternally indebted to Steve for his advice. Steve is the pioneer of the field of social genomics. There have been RFAs at NIH on the field of social genomics. And so it's now a more umbrella term for how do we understand how the exposome, social, psychological, behavioral factors get under the skin to influence the malleable aspects of the genome, the epigenome, gene expression, anything that's about the omes. So lots of people, yeah. So I mean, I think that we're all in awe of Steve. He's the most transdisciplinary thinker and scientist, and he reads everything. Basic science magazine articles don't get by him. I know if something's relevant, he's read it or sent it to colleagues. So Steve has also left academia. Actually, he never left, but he took a break to run Hope Labs and science program, research program. And it was just unbelievably remarkable and such a statement about his actual compassion for having an impact on mental health in the world, on youth. This was kind of a creative social entrepreneur organization that works on youth mental health and technology. And he did a lot of great studies. He helped fund the heart study while he was there. And I could just go on and on, but he basically is one of the most generous collaborators and brilliant thinking across levels from psychology to the omics broadly. And we are so grateful to have you and to collaborate with you and get to hear about what you're excited about now. So I didn't direct you in any way about what you're going to talk about, because I want to, we just want to hear what you want to talk about. We also love to be informal and have discussions. So we've got 50 minutes, and if you could save at least 10 minutes, and even more is better for us to ask you questions.
Sure, happy to do that. So thanks for the opportunity to talk with you guys, and thanks for that generous introduction there, Alyssa. As Alyssa mentioned, she gave me only hazy guidance about what I should talk about today. And so what I'm going to do is give a kind of a quick fire drill introduction into this field of social genomics that I work in and try to help people bring into their own lines of research. And then I'm going to spend a little bit of time talking about results from this breathing study that you guys had done, just to kind of help people understand how that work fits within the kind of broader schema of what's going on out there, what the similarities are, and how we make sense of genomic data in the context of psychobiological processes. And then, as Alyssa said, I think I'm going to try and leave as much time as I can to sort of answer questions. So first thing I'm going to do is share my screen, pop up some slides here. Let me know if you guys don't see this, but looks like it should be working. So yeah, I basically study the everyday life of the human genome, or the way that the life circumstances that we inhabit essentially outside us, or like our psychological, social, cultural, political, and economic existences, how all of that stuff outside us gets inside our body and regulates the activity of the human genome at a very concrete molecular level. So this is actually a strange thing for a genomics researcher to do. So generally, I begin my talks by just explaining how we got onto this topic in the first place. And it really came from one particular molecular epidemiologic study that I'll kind of show you here for a moment, that involved a very small number of people, in which we collected blood samples and surveyed the activity of every gene in the human genome in white blood cells from those individuals. And so in this study, this heat plot represents the essentially 200 or so genes that showed 30% or greater differences in their average transcription levels, the abundance of messenger RNA for these genes, in people exposed to high levels of the risk factor that this study was focusing on. So this study was done by an eminent social psychologist and psychobiologist, John Cacioppo, and he had these samples in the freezer, and he's asking me, can we use the genomic tricks that you guys had developed for analyzing viruses? Can we use that to analyze diseases that are mediated by human genomic function, not by viral genomic function? And I said, yeah, sure, we're happy to give it a try. And so this is what happened. It's like literally a freezer study where he just pulled out, it's like, I think it's like 5 or 6 people exposed to high levels of this risk factor, another 5 or 6 exposed to low levels of the risk factor. And each of the columns in this heat plot represents one of these 200 or so genes that emerged as showing these relatively robust differences in activity in the white blood cells of those people exposed to high levels of the risk factor. And to the extent that the cell defined by an individual study participant in a row and a column corresponding to a gene, to the extent that that's red, that just indicates high activity of that gene in that person's white blood cells. And so what these organized blocks of red and green are really telling us is there's about 70 or so genes that are systematically more active in the immune cells of people exposed to high levels of the risk factor, 130 or so systematically less active. And it turns out these are not a random smattering of all the genes in the 20,000 gene human genome. They actually represent a few kind of coherent themes. So for instance, among the genes that are systematically more active were a number involved in inflammation, which is the body's first line of defense against injury and sort of a major initiator of immune responses. And then prominent among the genes that are less active in people exposed to the risk factor was another block involved in defense against viral infections, including type 1 interferons that play a major role in innate antiviral responses, as well as immunoglobulin G antibodies. So, this kind of shift in what the immune system is doing with its molecular resources, I think, would not be particularly surprising if the risk factor that we were looking at were the kind of physical, chemical, or microbial things that we generally worry about. Epidemiology, things like benzene in the drinking water, cigarette smoke, or something. But what was interesting to me about this study is the fact that the risk factor that was structuring these differences in gene expression was simply the extent to which people in the first few rows of the heat plot felt disconnected from the rest of humanity. So it was loneliness that was structuring these differences in gene expression. And that was a little bit more of a puzzle for those of us who have traditionally thought about the immune system as, mostly involved in, sort of attending to microbes, and, maybe it has to pay some physiological debts or something like that. But basically, why should it be paying attention to your psychological or social life and, changing its molecular stripes as a result? So as we started to analyze this kind of thing in more detail, and other people started doing similar kinds of studies, we came to realize that this shift towards greater inflammatory gene expression and reduced antiviral gene expression isn't unique to loneliness. It turns out we see much the same thing anytime people are exposed to a sense of threat or uncertainty for more than, let's say, a few days or something like that. So anytime the human brain calculates our life as being threatening or unstable or uncertain prospects, it's just going to automatically turn on this sort of, you can think of it as molecular defense reflex. And we've learned over the last decade or so about how that happens. This is all mediated by fight or flight stress responses from the sympathetic division of the autonomic nervous system. So the neuroeffector molecules like norepinephrine that are released from those nerve fibers end up interacting with beta adrenergic receptors on the surface of white blood cells. And those receptors are essentially wired by millions of years of evolution to activate specific transcription factors that end up stimulating the transcription of pro-inflammatory genes like IL-1, IL-6, tumor necrosis factor, while at the same time reciprocally inhibiting the expression of genes involved in innate antiviral responses like type 1 interferons. So this kind of dynamic takes place largely in a relative minority population of the white blood cell pool, particularly monocytes and to a lesser extent neutrophils and dendritic cells. So these so-called myeloid lineage cells are interesting because actually they don't live very long in the body, and so they're constantly being regenerated by the bone marrow. And as a consequence of that, not only does this kind of sympathetically mediated neurobiological trimming of the immune cell transcriptome influence existing immune cells, but it actually also reshapes or recalibrates the immune cell regeneration process. So in adverse environments, the brain calculates, that we're under threat. It activates these sympathetic nerve fibers. In one place, these sympathetic fibers release norepinephrine is in the bone marrow microenvironment, where the hematopoietic stem cells are busily regenerating the white blood cell pool every day. In other words, if we lose probably trillions of sort of myeloid, certainly billions of myeloid lineage cells like monocytes and dendritic cells and neutrophils, then we need to replace them. And so that's what these stem cells are doing every day. And what happens when the fight or flight stress response releases norepinephrine is that ends up altering, let's see what's going on here. Suddenly, I'm not able to advance my slides, so I'm not sure. I got a message earlier saying someone had turned on some process, but I'm now, oh, there it goes. Okay, finally it's going again. So what happens is essentially these stem cells make more myeloid lineage progeny, monocytes and granulocytes that are born in the bone marrow, and then they make their way out of the bone marrow into circulating blood, where they essentially remodel the circulating white blood cell pool. So you've got more, particularly immature, highly pro-inflammatory monocytes, so-called classical monocytes, being born. And these cells then transit to particular sites of injury and kind of accidentally mess us up. So for instance, if we get a hit to the brain, these cells will traffic into wounded neural tissue and produce an unusually vigorous inflammatory response, which inadvertently leads to neurodegeneration. The same cells, when they're attempting to repair a tear in the coronary artery, end up more vigorously building an atherosclerotic plaque and promoting heart attacks. The same cells over-respond, producing hyperinflammatory responses in the airway, so you're more likely to get pneumonia and asthma. They also end up communicating with the lymphoid lineage cells, particularly T cells and B cells, and as a consequence of the signaling that they're biased to produce, they end up inviting those T cells to create immune responses that are good at clearing bacterial infections and other kinds of extracellular pathogens, but are less efficient at clearing intracellular pathogens that might require a cytotoxic T cell response. So things like a viral infection or cancer, also cytotoxic T cells are what we're looking for in that scenario. So, and last but not least, the same cells end up essentially inadvertently promoting the metastasis of solid tumors by executing a wound healing response when they ideally would have executed instead a kind of a cytotoxic or cytostatic response that would stop the tumor's growth. So in a wide variety of ways, this stress-driven remodeling of the white blood cell pool leaves us more vulnerable to sort of essentially a host of chronic illnesses. And what's happening essentially is the immune system, which is in social animals like ourselves, kind of programmed by default to respond vigorously to viral infections because they're these classic socially mediated pathogens, as we were all so recently reminded. But the problem is that the kind of immune response that's effective against viruses isn't very effective against wounding injuries and bacterial infections. So what seems to have happened is as our immune system sort of evolved over time to be more polarized by default in its resting state in responding to viral infections, it has retained the capacity to kind of pivot rapidly back to a more effective antibacterial response. And the way that pivot is triggered is when the fight or flight stress response gets read out by these adrenergic receptors. And that probably made sense when the things that triggered fight or flight stress responses were like that guy, which is sort of the stereotypical releaser for these kinds of physiological responses over most of our evolutionary history. But in the world we live in now, it's more stuff like this that triggers fight or flight stress responses. And these things aren't going to wound us being stuck in traffic or trying to get our grant funded or a book published or whatever. But even though they're not going to physically wound us, we still have this molecular machinery in our body that is executing this defensive pivot towards a more pro-inflammatory immune response during periods of fight or flight activity. So as a consequence of that, in the worlds that we live in now, stress and other adverse life circumstances end up being associated with greater cancer development, greater neurodegeneration, and greater atherosclerotic plaques. So, on some level, you can think of this as a bad recipe. It's sort of made sense once upon a time, the way we lived 20,000 years ago, but it's probably not so adaptive right now. But right now, it's literally taking unpleasant life circumstances and converting them into chronic illnesses. So what can we do about that? So, obviously from the, sort of the mechanistic research we've done, we have certain answers related to, yay, if we did, gene therapy on people and took this receptor out, or if we parked people on beta blockers all the time, yeah, we could, block many of these effects, but that's probably not a sort of the sustainable and productive solution. What we were interested in about 10 years ago is what can we do in the way we live as human beings that would change this kind of molecular biology. So when we're asking about how we should live instead of living in these adverse life circumstances, we're mostly talking about this topic with, for lack of a better term, happiness psychologists. And happiness psychologists said, yeah, we're happy to help you think about whether happiness will counteract this kind of adversity-related molecular toxicity, essentially. But you just have to tell us what kind of happiness you're interested in. I was like, what do you mean? What kind of happiness I'm interested in? Is there more than one kind of happiness? And they're like, yeah, we distinguish between hedonic well-being, the kind of happiness that comes from feeding your kind of self-interested consummatory reward system. So eating tasty food, seeing beautiful sights, watching funny movies, that stuff that produces happy moments. Kind of, you can think of it as the Epicurean model of a life well lived. But historically, as people have thought about happiness, and of course the people that think most about happiness are moral philosophers more than even contemporary happiness psychologists, they've often distinguished between hedonic well-being and a different kind of happiness that comes from what they would call pursuing virtue, trying to do something good for other people in the world, trying to kind of transcend your own immediate self-gratification and make contributions to, sort of a community, helping others, even if it's just a family or a business, or making contributions to, science, exploring the world, or art, sort of sharing psychological and spiritual, sort of experience. So as we were doing this kind of research, and I'm going to kind of skip through the measurement stuff since you guys are all familiar with that kind of stuff in principle. As we were doing this research, we bumped into a really striking disconnect where it turns out that people who have high levels of eudaimonic well-being, as you might expect, and as your grandmother probably would have told you, show relatively favorable gene expression profiles. They look great, low levels of inflammation, high levels of antiviral biology. But interestingly, people who express high levels of hedonic well-being, but comparatively low levels of eudaimonic well-being. In other words, they get to a happy place in life, but they're getting there not by this kind of contributory, pro-social, sort of value pursuit aspirational mode, but instead by just kind of feeding their happiness machine. Their biology actually looked terrible. They had high levels of inflammation and low levels of antibody response. And this was interesting because at the level of emotional states, these were two very similar groups. They had similarly high levels of positive affect, similarly low levels of anxiety and depression. So it didn't seem like these two types of well-being were, if you will, registering in the brain or conscious experience differently, but they were clearly leaving different fingerprints on the genome. And that turned out to be a surprisingly durable result. We see this not just in the North Carolina, college students that the study was originally done in, but in, Japanese salarymen working for a big IT company, Colorado video gamers hiding out in their dungeons playing, video games all day long, senior citizens living in Korea. It's a surprisingly robust phenomena that people who have high levels of eudaimonic well-being show relatively low levels of this kind of inflammatory gene expression profile. So we've spent a lot of time recently kind of thinking about how to get more eudaimonia into people's lives. So that initial observation, of course, is like an observational study. It doesn't tell us how to make more eudaimonia, but there's ideas that people have out there about how to do that. One thing I can tell you is that as we've kind of looked at this neurobiologically, we haven't found a eudaimonia hormone that seems to be mediating these effects. These effects really appear to be due to the fact that people who experience high levels of eudaimonic well-being seem to be pumping lower levels of fight or flight stress responses out into their peripheral physiology. And that is probably, at least in part, because the sort of aspirational reward seeking, hoping, wanting circuit in the brain, the so-called ventral striatum, and particularly the nucleus accumbens, a kind of key driver of, sort of identifying stuff you value and pursuing that stuff, trying to make it real in the world. This dopamine-rich circuit seems to be capable of laterally inhibiting the activity of threat responses. So if you've got an amygdala that is recognizing the world as threatening and getting ready to fire a fight or flight stress response out of the brainstem, it turns out the ventral striatum gets a vote there. And if the ventral striatum decides that you're doing what you should be doing, you're doing what's most important to you, you're pursuing what's most valuable, it will basically, not 100%, but quantitatively antagonize the activity of the threat circuitry that's actually driving this autonomic response that's shaping these gene expression profiles. And so you can see a surprising correlation between, for instance, just the resting level of pro-inflammatory gene expression in a person's white blood cell and how excited their ventral striatum gets when, in an fMRI study, you just present to people stimuli that they care about, as opposed to stimuli that they're not value invested in. So, real life protocols for trying to actually make this stuff happen. There's, as you might imagine, no magic recipe for creating a eudaimonic life, but some of the things that seem to produce that include a project that Teresa Seaman, a biodemographer at UCLA, has developed that basically involves getting grandmas out of their houses in South Central LA and into the local school systems to serve as teachers' aides. And so they show a really robust reduction in this threat-related molecular signature over the course of that 9-month thing. And this stuff is not easy. It's hard to take care of these kids that are so stressed out and often come from difficult home environments. But it's certainly rewarding, and it also builds a sense of community among the grandmas as they swap wisdom about, sort of how to be an effective teacher's aide. You can get similar kinds of results out of randomized controlled experiments where, for instance, Sonia Lubomirsky's group now has, in 2 separate studies, randomized people to perform random acts of kindness. And those who do that for a specific other living, breathing human being show significant reductions in this threat-related molecular signature. Interestingly, people who do that for the world in general, but not necessarily for another, living, breathing human being, don't show those benefits. And people who perform random acts of kindness for themselves actually, if anything, trend towards slightly higher levels of that threat-related signature, almost along the same lines as we saw with the people who had high levels of hedonic well-being, absent any kind of eudaimonic path for getting there. And of course, no change in control groups that aren't doing any random acts of kindness. So those are some examples of the kinds of things that we're looking at experimentally to try and make more eudaimonia in order to test whether there's a causal effect of this kind of eudaimonic well-being on these molecular threat signatures. We bumped into another interesting strategy for this kind of thing in a study where we were originally set up to be looking at loneliness and social isolation in monkey models. But we accidentally, it was quirky timing, we accidentally pulled this off right at the time that COVID lockdowns were coming to the fore, and there was a lot of interest in like, what is all of this isolation gonna be bad for our biology and our immunity? And so in this study, primatologist John Capitano at California National Primate Research Center would take adult male rhesus macaques out of their field cage where they live with usually about 80 other monkeys, and he would put them in their own little private monkey apartment. It was a pretty posh apartment as monkey apartments go. It had toys and plenty of food and everything. It was just isolated from all the other monkeys. And just as you would expect based on the story I told you earlier, if we look at those classical monocytes, we see them bump up in prevalence because the bone marrow is responding to threat and pumping out more classical monocytes. And at the same time, the sort of vigor of the type 1 interferon response, or its messenger RNA levels, is kind of down by 10 to 20% as a consequence. And this persists for a couple weeks at least. So after that, John put the monkeys back out of their monkey apartments and in the field cages, got them re-socialized. And then A month or two later, he locked them down again. But this time, when he locked them down, there was in the cage next door a little, infant monkey, basically. Not literally infant that like you had to hold, but like a probably a toddler age style monkey, cute little guy running around that even the adult male macaques thought, oh, this guy's pretty good. Let's, play with him and take care of him. And turns out that when you lock down the macaque and he's got somebody to take care of, somebody to care for, you don't really see either of those effects at all. In fact, you see a net trend downward in the production of those classical monocytes and no material decrease in the type 1 interferon response. So this notion of caregiving is interesting in part because in addition to these kinds of central nervous system level, sort of inhibitory processes that eudaimonia might have, there is a separate sort of fight or flight, rest and digest, sorry, not at all fight or flight, anti-fight or flight, rest and digest parasympathetic nervous system that, a branch of the autonomic nervous system essentially opposes fight or flight stress biology. And Steve Porges and others have theorized that, in fact, this, particularly the supradiaphragmatic wing of the vagus nerve gets especially active when people are giving care for others. This probably evolved for maternal infant bonding and nursing and that kind of stuff. But their argument is that this basic neural circuitry can be generalized from that particular context to your family or your business or some other, value-laden enterprise, and that you can develop this kind of parasympathetic, if you will, flow state just from, doing good stuff for others. So consistent with that, actually, we find that other indices of parasympathetic nervous system activity, like respiratory sinus arrhythmia, heart rate variability, is also correlated with most especially higher antiviral biology. We see sometimes associations with lower inflammatory gene expression, but that's more sporadic. But for whatever reason, the respiratory sinus arrhythmia, this index of parasympathetic activity, seems to be particularly tightly linked to the sort of antiviral, if you will, tone of the circulating pool. So I don't want to leave you with the idea that eudaimonia is the only thing that can counteract these effects. Like I said earlier, we can deploy medications to block these effects. Pharmacologic antagonists of beta-2 adrenergic receptors work pretty well in that regard. Wellness practices like meditation, tai chi, stress reduction, that kind of thing, excuse me, also work for this kind of thing. But, doing good stuff for the world is a kind of an interesting option when we think about how we might go about, making a better life for us to live. So one of the reasons we are so interested in identifying different ways to be, sort of, if you will, counter CTRA, defeat threat, is because one thing we've noticed is that any of these interventions seems to work for some people, but not everybody. So meditation is a classic example. Some people love to meditate. They show these great stress reduction effects. Wonderful. Other people can't stand to meditate. They don't do it. And as a result, they don't show any of the stress reduction molecular benefits. So we've gotten really interested in trying to learn what works for each specific individual, not what works for people on average. So essentially not ask the question, how should we live in general as generic humans, but how should one specific human being live? And the way that we go about answering this is by essentially recording how variations in everyday life circumstances affect variations in these threat-related molecular signatures. So the idea is we're going to take individuals and repeatedly sample over time this kind of molecular monitoring process, and they kind of essentially bounce that information out of their body and kind of process it in the outside world. The ultimate kind of vision we have is that somehow we'll have little micro sensors in the body and bounce things into the cloud and then feed them back stuff through their digital apps and stuff like that. But as you might imagine, there's a lot of opportunity in this kind of thing. You could potentially use this in the context of medicine to detect disease, promoting physiological regimes before they actually produce disease. You could use it for epidemiology or policy analysis to kind of get a sense of kind of at the biomarker level, how are large groups of people doing. But the most powerful opportunity, we think, is actually to ask each individual what is the best way for them to live in the sense of sort of molecularly quantified self, or a sense of molecular self-awareness that would allow you to essentially use that as biofeedback for tailoring your life. So I'll show you an example of this where we take sort of a multi-gene composite score measuring that threat-related molecular signature, this conserved transcriptional response to adversity. We flip it on its head so that it's scored as well-being. And then we have people keep track of what's going on in their everyday life. So here's one person who did that kind of thing. She was unusually vigorous in the number of channels that she was recording from in her everyday life. But we've got all of these. You can think of them as candidate streams of information about what's going on in life. And we can feed that to machine learning algorithms to try and give us a sense of, sort of what amongst all of these streams actually best accounts for the pattern of, molecular well-being versus ill-being that we see in this individual. So for instance, in this particular case, what popped up for this young woman was, her molecular well-being scores were high, her CTRA levels were low on days when she felt that what she was eating was healthy. Who knows exactly what that means, but she's got an idea about what a healthy diet is. And when she ate a healthy diet, her molecular well-being looks pretty good. The more time she spent outside, the better. The more intense exercise she got, somewhat the worse. That's not a surprise because we know intense exercise is somewhat pro-inflammatory. So there's probably a little kind of click in that direction. It's not clear that that means that she shouldn't exercise. It just means that there is a kind of molecular toll to exercise. When she was, quote, socializing, she also showed lower levels of molecular well-being. That's probably because she had distinct channels for things like spending time with friends or family members and other kinds of things like that. So for her, socializing really came out to be partying in the dorm that she lives in. So you can imagine how that might be a little bit of a setback for your molecular well-being. And then she was collecting data during the summer when she worked in a kind of a business setting where it was intensely competitive. She was basically a commission salesperson. She was very existentially miserable at the idea that the only way she could make any money is by essentially stealing it from somebody else. So that was like a summer of misery for her. The more time she spent at work during that miserable summer, the worse her molecular well-being looked. We can also do this kind of thing for the distinct channels of molecular biology in the composite signature. We can look at inflammatory genes and see how they wax and wane in activity, and interferon gene expression. We can ask whether different features in this person's life stream might correlate differently with these two different types of molecular biology. And in fact, it turns out what matters for inflammation in terms of lifestyle circumstances is to some extent substantially different than what matters for the type 1 interferons. So I won't take you through the fine-grained details too much, but there's some degree of overlap here. Sleep matters for both of them. The daily positivity versus negativity matters for both of them. And even eating fat matters for both of them. But mindfulness matters a lot for inflammation, not so much for type 1 interferon, so on and so forth. So this is really all just kind of an illustration of this idea that in addition to doing these sort of group comparison studies that are sort of our dominant trial design, we might start thinking about these intra-individual sort of N of 1 followed over time paradigms to give us ideas about how to change people's lives. So far, of course, we're doing this only in a kind of an observational or monitoring sense, but I think our long-term interest is to give people this kind of molecular self-awareness so that they can actually start to make changes in the way they lead their lives. And of course, the ultimate proof of the merit of this or lack thereof is whether that results in changes in these molecular threat signatures over time, which presumably will reduce the individual's risk of those chronic diseases that those cells contribute to in the first place. So, I mentioned before parasympathetic nervous system activity and other things that are, you can think of them as sort of lifestyle approaches to reducing stress biology. So I thought I would drop in here just some summary of what we learned from the STAR study, which you guys are all more familiar with than I am. I'm sort of vaguely acquainted with the hypotheses, but you guys have, lived it and breathed it, literally, I guess. So in this study, you guys are interested in breath and especially this sort of Wim Hof sort of crazy, I don't know, hypoxic breathing protocol. It sounds kind of insane to me every time Alyssa describes it to me, but whatever, it seems like reasonable science. So I'm always happy to be of help. So you guys had a number of general questions that you were interested in. One is like, is this hypoxic breathing going to change basal gene expression profiles in the circulating white blood cell pool? And you guys had some specific hypotheses about what might happen. And those hypotheses worked out pretty well, actually. So compared to the control group, those who were in the hypoxic breathing group showed reductions in inflammatory gene regulation, particularly genes that are regulated by the pro-inflammatory transcription factor NF-kappa B, increases in hypoxic gene regulation, sort of expression of genes that are controlled by HIF1 or HIF2 alpha. We also saw increases in DNA damage repair processes, which was interesting. You guys kind of called that. I was like, yeah, this doesn't make any sense to me, but you guys were right. So no problem on that. And then increases in sympathetic nervous system related signaling, probably because the hypoxic breathing kicks off various kinds of autonomic responses that are registering as sort of threats or alarms. So this doesn't seem to be terribly toxic to the body. It doesn't seem to be flowing through and driving more inflammatory signaling. In fact, it's associated with less inflammatory signaling. But just for what it's worth, there does seem to be an autonomic response component to this hypoxic breathing. And all of these effects are, robust to control for various, both, time varying things in the pre to post protocol world, as well as for demographic characteristics. The second question that you guys had involved this acute stress challenge study where you guys were basically, I guess, triggering people and then asking, how does molecular biology respond to that? And does hypoxic breathing change the nature of that molecular response to this model acute stressor? So what we saw, a couple of, moderately, predictable or promising results. One was we know that this Trier type stressor activates the sympathetic nervous system quite efficiently. So that generally is associated with increased expression of genes that are regulated by the CREB transcription factor, which carries signals from the beta adrenergic receptor into the transcriptional regime. So that part made sense. It did appear that the hypoxic breathing protocol was somehow essentially buffering the body against this sort of acute stress-induced response through this beta adrenergic signaling pathway. We saw a marginally significant decrease in the stress-induced response by the hypoxia-induced transcription factor HIF. But it was a little bit of a quirky result in the sense that if we controlled for changes in the relative prevalence of various white blood cell subsets, that effect went away. So it may be due in part to differences in HIF expression just across different leukocyte subsets, and then variations in how those leukocyte subsets get mobilized during these kinds of acute stress experiences, which is kind of a well-known phenomenon. We didn't see any change in NF-kappa B or DNA damage repair pathways. That said, this was a pretty challenging little study to see those molecular responses because we only had relatively short duration, kind of post of the acute stressor. So if there were effects that occurred at later time points or were sort of like more chronically present in the basal gene expression profiles, they would not have appeared in this. And then the other caveat being there seems to be something related to the HIF results that are related to cell redistribution. The third thing was a question about just if we have people do the hypoxic breathing protocol right there in the lab, what happens to them? And interestingly enough, the people that had sort of, you can think of them as chronic or, essentially, I guess, chronic hypoxic breathing, when they did that in the lab, there was an interesting reduction in HIF1 activity and NF-kappa B activity that took place. So very acutely, the effects seem to be different than this chronic impact on the basal leukocyte transcriptome. And then people who, did these hypoxic breathing protocols showed significant declines in CREB activity and increases in P53 activity, which is this DNA damage response during the paced breathing task. So In other words, everybody who's doing this sort of hypoxic breathing protocol seems to show a significant decline acutely, very instantaneously, relatively speaking, in CREB-mediated gene transcription, increases in p53. But again, controlling for leukocyte subset changes seems to mitigate some of these effects. So that's what I got for you guys today. Once upon a time, we would always end talks with this almost facetious statement that, hey, watch what you're doing with your life. The way you live your life today is going to influence the molecular composition of your body for the next 2 to 3 months. Or if you kick off a positive feedback cycle, maybe it's going to change you for the rest of your life. So we used to tell people, hey, don't mess up. But now, we can actually do this kind of stuff and measure how these kinds of things are impacting an individual's molecular well-being and whether they're in some sense living life the way they were meant to. So this idea of asking the human genome whether we're living the good life, seems like kind of an odd way to attack these problems. But on the other hand, with millions of years of experience helping us survive and thrive in this world, the human genome may have learned a thing or two about how best for us to be in this life. So with that, let me thank you guys and answer any questions that you have. Can anybody hear me? Somebody put a thumbs up or something like that so I can at least know. Okay, thank you, sir. Much appreciated.
Can we just jump in with questions or
Yeah.
Hello, thank you very much for the very interesting talk. I had a quick question regarding the expression of the genes related to ammonia, and in specifically, what are your thoughts on whether these are on the role that maybe regional expression of these genes could play in distinct brain regions? Like, would you, given that specific brain regions are involved in autonomic control, like the anterior cingulate,
Yep.
the insula, whether you have any thoughts of this being more of an organic thing happening in the whole body, or I'm more curious specifically about your thoughts though in relation to regional brain expression.
Yeah. So the simple short answer is I'm agnostic, just because in general I don't believe that gene expression dynamics in white blood cells that we're able to see in that study really have much of an implication for what's going on in the brain. I do believe that stuff is going on in the brain. I just don't believe that we can actually know what it is very reliably until we get some brain tissue out. So with that said, we've actually started doing brain studies. I kept asking people for their brains and they wouldn't give them to me. So now we're mostly doing mouse studies where we use chemogenetic techniques to activate that same sort of seeking, hoping, wanting ventral striatum system. And then looking at both how the peripheral immune system responds to that kind of thing, and then trying to track down what might be going on in these specific regions of the brain. So I do believe that there's likely to be, significant changes in some of these ventral striatal structures, particularly the nucleus accumbens. The exact nature of those, I don't know, and I'm certainly not the only person looking at this. So in addition to our project, Asia Rolls at the Technion in Israel is doing similar kinds of things, and so is, who else? There's one other group doing this kind of thing as well that I'm kind of skipping on at the moment. But to make a long story short, I would say it's very likely that there are changes at the brain level, but it's not clear to us at the moment exactly what those are. Do they have to do with increasing, neurotransmitter efficiency and those kinds of ventral striatal structures, or do you get hypertrophy? Oh, it's I know that the other person is there's another neuroscience group in Israel that is using rats, and they actually have the rats help other rats who are suffering. And when they do that kind of helpful rat paradigm, they too find changes in nucleus accumbens gene expression, the nature of which are hard to summarize. There's quite a lot of molecules behaving differently, but it's difficult to know whether that is due to general, sort of hypertrophic activity, or whether that is more selective to a particular neurotransmitter system, effector system. So to be determined, but great question.
Can I follow up with would your expectation be that the same genes are involved, or
No,
distinct classes
my
of genes?
expectation is that absolutely different genes are involved. And that's because everything I've learned as an RNA biologist is that RNA is very, very local to the cells that are being sampled. And so, immune cells have a completely different, you can think of it as life history strategy than neurons. They stimulate, themselves differently. They have different, sort of things they're supposed to accomplish in the world. The kind of way they bring information in, the way they respond to it is pretty different. So my assumption is that it would be qualitatively different, just simply because, the nucleus accumbens is not out there mostly trying to find microbes to destroy. It's mostly out there trying to compute stuff about how to act in the world to advance your interests.
Steve, thank you so much. You're not done yet, but it was a social neutrality experiment condition you were in, because when you finished, there was silence. It's because we were all muted. We were like giving you the hugest applause. So,
well, thanks for letting me know that. I'll take any appreciation I can get.
A little post-hoc, but you should be ringing it in your ears. It's an amazing line of research, and especially how you answer your questions in so many different models: rodents, humans, observational intervention. I mean, the list has grown. When you first, showed the eudaimonic versus hedonic finding, it looked like it could be a fragile finding, and you have just taken the time to replicate it so many times. I couldn't even count how many citations.
Well, yeah, I think that's because I remember having a walk around the streets of San Francisco with somebody who was kind of thinking about all of these things, and her advice to me was, you can argue about it all you want, but the best thing to do is just replicate and, let the chips fall where they may and the evidence move on despite the controversies.
Yeah.
So good advice there, Dr. Apple.
Oh, I was wondering, I was like, his memory's better than mine. I bet that was me. So Steve, we're so excited that you, but also you have to have the stress resilience. If you're going to enter dialogue with pigs and monsters, you have to have a lot of sturdiness.
Yep.
So congratulations to your team. I think I would have melted, but the data in the end won, which is the best thing. Okay, so you spent a lot of time on our STARS intervention, and yes, it's precious data to us. We're very excited about it. Two sad updates. We lost the serum, right? You knew that, right? We lost all the serum we were about to send you. So we now lost the active cytokine question and other interesting things like GDEV15. So we're
you lost it as in your freezer crashed?
I don't even have words for the trauma. Just,
Yes. And we can, once we stop recording, we can tell you all.
Yeah.
Oh, okay. So sorry for not calculating out all the contingencies here, but yes, I'm happy to hear about that later. And
we've been yelled at for not having double samples of everything and storing them in double freezers and this and that.
that is kind of excessive, actually. The bigger thing I worry about, do you still have the physical samples?
So here's the thing. First of all, we're so grateful that you got that done, that you got the sample in time, but we have cells and we have frozen PBMCs, and we are going to do single cell RNA sequencing. And so some of the questions you have, like, is it cell redistribution? We can answer. So that is going to salvage our, pick ourselves up from the ground here. It's like that level of analysis will be helpful, even though we don't have the active serum.
Yep.
But I have one question for you, and Ryan probably does too, and I'll just be really quick. You showed in many studies that CREB, downregulation of CREB-mediated gene expression is one of the most common pathways of all the forms of distress. In the STARS intervention, which is positive, we think it's a model of positive hormetic stress, you showed the basal upregulation of CREB. At baseline, there was more CREB-related gene activity. Right?
Yeah, the first part of your question, I'm not sure is right. In general, what we see is that people who are exposed to threat or adversity or stress show higher CREB activity because there's more beta adrenergic signaling.
Okay, so this fits well with what you see.
Yours does fit well, yes.
Do relaxing, mindfulness, meditation, etc. Okay, so maybe we don't have.
Yeah,
thought-
and so the basal results that you have look great, honestly. Like the AIM-1 stuff is just, you know, what I think any straightforward, you know, hoped for result would be. The AIM-2 results and the AIM-3 results are, you know, more complex. Some of the stuff makes sense. Some of the stuff seems intuitively backward. But in both cases, it's just harder to interpret those things because they are probe tasks. They're not basal, sort of walking around native state stuff. And the kind of redistribution effects, play havoc with bulk sequencing data. And so that, is one issue to kind of take into account, but also the temporal stuff. So there's, acute stresses often don't have much of an effect on gene expression because, the system is generally engineered to be robust. And so it's not supposed to be super twitchy. It's supposed to adapt to change in lifestyle, but not necessarily a change in mood, if you will. So I think that's the other thing to bear in mind is it's just a taller order to try and get the other two AIMS results, to work out in any predictable way. But you've got results, so regardless, something happened, and it's certainly legit to report that and interpret it and think about how you would make use of it going forward.
Thank you so much. That's super helpful. Any questions from the room? Any questions from the Zoom audience?
I have a lot of thoughts going, so thanks for going through this overview and then going into the STARS piece more specifically, and especially in isolating the time piece. I think that has been really helpful to better set like our expectations of when we're going to see the acute versus basal. I was wondering if you could just speak to the P53 finding in that AIM-1 a little bit. I remember you talking about it being counterintuitive to you initially, and I'm just curious if you could talk through that piece again.
Yeah, I mean, DNA damage responses are fundamentally driven by DNA damage, and so I think you guys were saying, well, it makes sense to us that hypoxic breathing is potentially gonna decrease DNA damage. I can't remember what your original hypothesis was, but idea. So essentially, I think the hypothesis, if I remember correctly, that you guys had was that that P53 would upregulate based on virtue. Like it's like a protective, you can think of it as prophylactic physiological response. Whereas in most of the ways that I've seen cell biologists talk about P53, they really think of it as a reactive response. In other words, it only gets activated when the DNA policemen detect genomic damage. And so in that case, P53 isn't a kind of a virtuous prophylactic. It's actually a sort of a sign that something bad has happened to the genome. So my kind of interpretation of a P53 result would be a little bit, potentially different from yours if you're thinking about it that way. But even more so, I was like, nothing that they're doing in hypoxic breathing is damaging the genome as far as I would expect. And so I was like, I was thinking like that, I would expect nothing to happen here. And so the fact that you got the effect that you got in the direction that you predicted, I thought was, pretty impressive. So I think the only issue is to just like include that interpretive caveat in your discussion as you're presenting this stuff. But there's no reason that should talk you out of owning the hypothesis that you guys had in the first place. Like you guys clearly theorized, DNA damage response is a salutary prophylactic, physiology protecting response that we think this hypoxic breathing is going to induce. That was your hypothesis. Great. Write your introduction about that. The only time you have to talk about any potential alternative biological interpretation is in the discussion.
Perfect. Thanks for going through that.
Sure.
Thank you
Anything
so
else, you
much.
guys?
We get to talk more about that study. But we are so great. It's past 1, so we should let you and everyone have their lunch. But we are so grateful for your time chatting with us and that amazing tour of the world of your work, which is really a whole field. And just grateful for your generosity in mentoring many people. Ryan, the most recent from UCSF, but you have quite a legacy. So
Well, that's good. I'm glad it's helpful. I'm glad it all made reasonable sense. Thanks for the opportunity to chat with you guys and to continue collaborating with you. Keep up the good work and be back in touch whenever I can be of help. Okay.
Yes, absolutely.